Dr. Palmer, could you state your full name for the record, please?
Michael Palmer.
And do you promise to tell the truth, the whole truth, and nothing but the truth?
I do.
Thank you. Dr. Palmer, I understand, and I'll just review a little bit of your background. You are you have your medical degree, which you obtained in Germany. Back in 1991, I think, and you are you have extensive experience in pharmacology, biochemistry, medical education. You have a substantial background in medical research and teaching, a particular focus on drug mechanisms, toxicology, and the biochemical basis of disease. You've contributed to the scientific understanding of the mRNA vaccine technologies and their implications. I understand that you were a former associate professor of biochemistry at the University of Waterloo. You're a published author in many peer-reviewed medical journals. Your areas of expertise can be summarized as clinical and molecular pharmacology, drug metabolism and toxicity, mRNA vaccine mechanisms and safety profiles, and medical ethics and healthcare policy. Does that?
Well, healthcare policy not so much, but on the other hand, I actually also have a specialization in medical microbiology. So while I was still in Germany between 1991 and 2001, I spent 10 years at the University of Mainz in the Department of Medical Microbiology, doing laboratory diagnostics of infectious disease and also research on microbial pathogenicity.
Okay. And you came to Canada when then?
That was 2001.
And very briefly then, your career path from the time you came to Canada, I know you ended up at the University of Waterloo and then had to leave.
That's right. I joined the university immediately in 2001 and stayed there until they fired me in 2022 for refusing the COVID shot. That was
You and a few other witnesses at this inquiry over the last 2 years with similar similar history. I understand that. Well, why don't we just deal with that a little bit before we get into your PowerPoint, because your personal history is of interest to us. Tell us what happened.
Well, good. I mean, so I think they really wanted to get rid of me, and I think they used this vaccine mandate. They kept it just long enough that they managed to get rid of me. 5 weeks after they had gotten rid of me, they rescinded the vaccine mandate, which at the time had already been repealed by the province. So it was really kept in place for a few additional weeks by the school. And the reason why they would want to get rid of me would have been, I assume, that I had written a number of open letters. I'd also collected signatures from workers, students, faculty colleagues at the school in opposition to the vaccine mandates.
So you had begun to express counter-narrative,
that's right, yes,
expert opinions on the safety and efficacy of the vaccine.
Yes, that's right. I mean, the vaccine mandate was instituted sometime in summer 2021. And at the time, the vaccinations had already been going on for more than half a year, and there was plenty of evidence that the vaccines were very harmful. And I mean, I collected some references from the literature and incorporated those into the open letters. It was also clear already during 2020, which I also pointed out to the university leadership, that there was really no need for the vaccinations because COVID really wasn't a real killer virus after all. I mean, the infection fatality rates were no higher than they would normally be during an influenza pandemic, during which nobody seems to find it necessary to impose some mandatory vaccinations.
Right. And we're going to get to your PowerPoint in a minute, but when you were in effect sounding the alarm to some degree on the inappropriateness of the mandates and
yes,
perhaps on the exaggeration of the risk of COVID, which led to your abrupt departure from the university.
Well,
as
the
you've
abrupt departure was really occasioned by my personal refusal to be injected.
Right. But prior to that, you had already aired your views
Oh yeah, absolutely.
about
Yeah.
this.
Yeah, yeah.
This and were you getting any response, substantive response, from though I don't know who you were communicating your concerns about
Well,
with, but were you getting any substantive response from the public health authorities or anyone else to your concerns at the time?
no, I mean, I directed my open letters to the university leadership because they had imposed a mandate on all the university employees and so on, and the students. And I never got a direct reply from anybody there. I mean, it was all through the newspapers. So the newspapers actually wrote a couple of articles about this. And then they interviewed the leadership as well, and then they would always say, okay, yes, we distance ourselves from this, and so on and so
So were
forth.
you getting, to some degree, disparaged or demeaned by public response to your concerns? Did you get the sense that you were being targeted in any way? I'm just asking because we've heard backgrounds from other witnesses to that effect. Before we get to your PowerPoint.
It didn't really leave a strong impression with me, so.
All right.
There was nothing substantial in those
All right.
replies,
Okay.
yeah.
All right. Well, let's now, if you're ready and have it set up, do you want to start
okay.
your PowerPoint?
Yeah,
then?
sure. I'd like to. Yeah, thank you. Okay, so thank you for the opportunity first off, and my subject today is why no mRNA vaccine will ever be safe for children. Okay, so this what the experience that we have collected with the COVID vaccines must lead us to expect similar levels of toxicity with any and all future mRNA vaccines that might be introduced to protect anybody from other virus infections. Could be childhood diseases, could be influenza, could be RSV and whatnot. Regardless of what the specific antigen would be, we must expect the same sort of thing with the with any and all future vaccines. So why do I make this case? It really comes down to the question that the observed toxicity must be attributed to the properties of the mRNA vaccine platform as such. It is not predominantly due to the toxic properties of the spike protein, even though that is a toxic protein. It does have harmful effects, and it may play some role also in the observed side effects, but I think there is good reason to believe that the main pathogenicity, the harm done by the COVID mRNA vaccines is mainly due to the inherent properties of the mRNA platform themselves, itself. Okay, so, right. So you have seen data such as this before, I guess, so I won't spend too much time, but these are some numbers I just pulled up from openvs.com, which is a citizen-operated website in the United States that tracks the data of the VS database, and these are just the side effects observed or reported to the VS database of the COVID vaccines. And so what we really see in the graph here is the total number of severe adverse events reported to VS during these indicated timeframes, and we see a very steep rise in 2021 when these COVID vaccines were introduced, a steep drop afterwards because people stopped accepting those vaccines. And the numbers on the left, they all pertain exclusively to the COVID vaccine. So we have among other things, 200 deaths reported to VS due to the COVID vaccines. And just for context, okay, here we have, these are data that apply not only to children, but these are all ages, including children. And this is the relative risk of death, life-threatening events, and hospitalization due to COVID vaccines calculated per injection relative to the entirety of all flu vaccines. And we see that in every category, the risk of such an event is more than 150 times greater with the COVID vaccines than with the flu vaccines, which is really a disastrous safety record. But you know these numbers, we don't have to dwell too much on them. just for context, so in the past, vaccines were withdrawn for a lot less damage that they had done. For example, there was a rotavirus vaccine once upon a time, and after about 1 million doses of that vaccine had been dispensed, 15 cases of interception had been reported. So that's a complication that arises from some mishap in the large intestine. It is a dangerous condition, but a treatable one if it's diagnosed properly in time. But nevertheless, after these 15 cases, this vaccine was pulled. And we have seen much greater numbers of harm with these COVID vaccines, and they have not been pulled. And this is not just in the United States, this is internationally. So I just want to point out, obviously something really changed in this area. There must have been some political influence that caused such a withdrawal not to be even discussed, really, right? So there is really something very concerning behind the scenes going on. Okay, so in order to make the case that indeed all future mRNA vaccines will be harmful, I will first review a little bit of underlying virology and immunology. I will try to keep it as simple as possible. So here we see a simplified sketch of how a virus multiplies within human cells. A virus particle, at the very minimum, contains a nucleic acid in the middle, the nucleic acid genome, and it is encapsulated by a protein shell. There can be a membrane around as well, but here we have a simple virus with only a protein shell, which is shown in green. Once the virus binds to some receptor on the cell surface and is then taken up, it undergoes uncoating, which means that the nucleic acid is released within the cell. And the nucleic acid contains the blueprints for all the viral proteins, including the ones that will form the shell of the progeny viruses. The machinery for turning the information in these blueprints into actual proteins is supplied by the cells. These are the little gray blobs here, the ribosomes. We have some viral proteins which will become part of the progeny virus particles. We have other viral proteins which carry out functions within the cell, such as, for example, the creation of additional nucleic acid molecules, which then will also be incorporated into the progeny virus particles. Now here for illustration, we have a cell highlighted in blue, which has produced tons and tons of little red virus progeny particles. So a virus-infected cell can become a veritable factory of progeny particles, and it is clear this cell is gone, so to speak. So the only way to deal with such virus-infected cell by the immune system, for the immune system, is to kill off those cells. So that's a key aspect of immunological defense against virus infection. It consists in killing off the virus-infected cells. Now, the immune system uses, among other things, antibodies to suppress virus infections. How do these antibodies work? Essentially, they have two effects. One of them is to bind and block the virus particles before they can even enter and infect a cell. If that happens, this means if we already have antibodies before we encounter the virus, then this is the likely outcome. The antibodies will bind and neutralize the virus particles and nothing further happens, which means we won't get sick. We are immune. Now, if we do not have antibodies yet at the time we encounter the virus, then the virus will get its chance to infect some cells and will start multiplying. And then the viral proteins will be made inside the cell. And then antibodies, once they are formed, right, once these viral proteins start showing up, the immune system starts making antibodies against them. Once the antibodies are available, they will bind to the proteins on the surface of those infected cells, and this will set in motion several mechanisms for killing those cells. One of them is illustrated here. It's the activation of the so-called complement system, a cascade of serum proteins that will then form holes in the cell membrane and will actually destroy the cell that way. There are other mechanisms, but we don't have to go through all of them. But I do want to mention the T killer cells, which are another prominent mechanism for antiviral defense. Here again, we start with the viral protein, which is made after the virus infection of the cell. Some of these protein molecules will undergo fragmentation by the cell within the cell. That's a service supplied by the cell itself. And the cell then also takes some of these fragments to the cell surface, where they can be recognized by T killer cells. The T killer cells will take this as a signal that this cell is infected and needs to be destroyed, and will then inflict some cytotoxic action on the cell and kill it off. Okay, so now the idea of vaccination. What should a vaccine do? It should actually endow us with antibodies, which should already be there, should be already available before we even encounter the virus. So the antibodies should get there first, and therefore the virus infection should be suppressed. Okay, so how this requires that we find a way to acquaint our immune system with the virus in question, and there are several types of vaccines that can be used for the purpose. The most straightforward type of vaccine is the inactivated virus vaccine. For example, the Salk polio vaccine, the first polio vaccine, was one of those. You prepare it by growing up the virus in question in cell culture in large volumes, and then you treat the virus particles with some reactive chemical, which will destroy its ability to multiply, but it will retain its ability to stimulate the immune system to form antibodies. So all you have to do is purify those inactivated virus particles and then inject them, and you will get your immune response. Then in a more sophisticated type of vaccine is the live virus vaccine. So this is an actual virus, but it has been mutated. The mutations, at least traditionally, were simply introduced by serial passage. So you just simply grow up the virus in cell culture, take the virus, grow it up again in another batch of cell culture, and so on. And after enough of these serial passages, the viruses will likely have accumulated enough mutations that will that will reduce their virulence for humans. So you can take these so-called attenuated viruses, inject them into patients, and then expect that they will develop only a mild and typically local infection. Of course, all of these vaccines have their own risks and drawbacks and complications. We won't go into those. We will simply note that risky these vaccines are, right? Their risk is eclipsed by far by the mRNA vaccines, which have been introduced recently. These superficially resemble a live virus vaccine. So again, we have a nucleic acid which is introduced into the cell, and the nucleic acid is then information in the nucleic acid is then turned into the actual protein antigen. In the case of the COVID vaccines, this would be the famous spike protein. Now, the mechanism for introducing this mRNA, in this case, into the target cells is not a natural virus capsid, but it is just a synthetic shell of lipid molecules. And the key difference, one key difference, is that this shell of synthetic lipid molecules has no immunological similarity with the proteins encoded by the mRNA, and we will see why this is important. So superficially, we could conclude, right, because the mRNA vaccines are similar in a way to live virus vaccines, that they should also not be any more dangerous. And this is indeed what is suggested to the public by the official propaganda. But it is wrong for several reasons, and I have listed them here. First off, the lipid nanoparticles which encase the mRNA in those vaccines have very low tissue specificity. They can essentially infect any and all cells, including the cells in the blood vessels. In the endothelial cell layer, the innermost cell layer of the blood vessels. And then these cells, after they express the viral antigen, spike protein in this case, they will be attacked by the immune system and destroyed. And the damage done to the blood vessel walls in this way is what is the main stimulus, the main trigger for blood clotting. Okay? So you have heard maybe about the D-dimer being increased in many vaccinated persons. That is just a laboratory manifestation, but in enough people we also have observed stroke and heart attack and this sort of thing. That's the key mechanism by which that is triggered. Then another point is that the mRNA vaccines, at least the ones that have been used so far, do not replicate in our body cells. So in order to induce the production of enough antigen for triggering an efficient immune response, you actually have to inject a large amount of this mRNA all in one batch. With a virus, with a live virus vaccine, you can inject a small amount and then simply wait for it to replicate in vivo. If that person already has immunity, the virus simply won't grow, so nothing much will happen. With mRNA vaccines, these things will actually have to be injected in a large amount straight away. And then on top of that, these particles, they sort of fly under the immune system's radar, and I already alluded to this. Right, with a natural virus, we have two possibilities. We either have antibodies to begin with or not. If we do have antibodies, then the antibody will already block the virus particle before much else happens. If we don't have antibodies, the cells will make viral proteins, will become infected and make viral proteins, and then we will have antibodies, and the antibodies will limit this process, this viral proliferation, as soon as enough of them are there. So in a sense, it's a self-limiting process. With the mRNA vaccines, if we already have a high level of immunity, maybe because we already got another vaccine dose or we have the natural infection already in our history, then the antibodies are there, but they cannot recognize these lipid particles, which means that the lipid nanoparticles will be taken up by the cells of our body. These cells will then make the protein antigen, and it is only then that the antibodies will actually be able to recognize those cells. And what they will do, the antibodies will then cause attack of the immune system on the cell that expresses the virus protein. So in a nutshell, if you have pre-existing immunity with a natural virus, it suppresses the damage. If you have immunity against the antigen encoded by an mRNA vaccine, it increases the damage. It amplifies the damage. And this is what we see, for example, in the statistics on myocarditis. Here are again data from the VS database. So we have data for both the Pfizer vaccine and the Moderna vaccine. They are really quite similar, right? We have some myocarditis cases after the first injection, but we have many more myocarditis cases after the second injection. And this is because the first injection induces immunity. The immunity is then already there. The immune system is activated. Antibodies are present at the time the second injection is administered, and then these antibodies can immediately pounce on those cells that, after the second injection, start expressing the viral antigen. So the immunity induced by the first injection sets the stage for the severe myocarditis after the second injection. And this is, sorry?
I was just going to wonder the time frame, and what does this particular chart, what period of time and perhaps demographics does this chart capture?
Okay, so yeah, that's a good question. So I mean, we see here on the x-axis the day after injection, right? So the myocarditis gets diagnosed in most cases within the first week, right? You can see that here, and that goes for both the first and the second injection. The injections are timed 3+ weeks apart, right? So the recommendation is to administer the second injection between 3 and 6 weeks after the first injection, and this would be enough time for the first injection to have taken full effect and to have induced the antibody response. So that is a perfect setup in a way, right, for maximizing the damage.
And it shows on the left the number of cases, reported cases.
These are cases reported to the VS database. And I mean the demographics, so these are mostly young men.
Right.
Okay, there are also young women, but it is firstly a preponderance of men or boys over women or girls. And on the other hand, also the age is typically, it's mostly the highest incidences in the between 20 and 30 years in the VS database.
Roughly when might this information that being available indicating what these graphs are showing us? Is this from
Okay.
early in the rollout of this?
So this particular study is from 2022, but
Okay.
the data started piling in immediately. So there was
Right.
essentially enough evidence on myocarditis already several months after the rollout. So in particular, there was enough evidence on myocarditis before the universities and schools across Canada instituted their mandates.
And this ties in as proof of the establishing a causal relationship between myocarditis and those other. In the prior slide, you indicated 3 areas of damage caused by the mRNA, and myocarditis just being an example of, I think, the first item on that list of 3.
It is, yeah, I mean, this is just one. I mean, damage can occur in very many different organs, and I will actually show you a few more examples
Okay.
down the road. But myocarditis is an important one, certainly, because the heart is a pretty important organ, and also, I mean, it doesn't really repair damage, right? I mean, if you have liver damage, the liver has a high capacity for regeneration, but heart muscle cells, once dead, are dead, so they remain dead. They cannot be repaired. They can only
Right.
heal by forming scar tissue.
Okay.
Okay, so the key aspect, the key point I really want to make here is that the damage is much more intense after the second injection. Interestingly enough, actually, it does drop off after the third injection, so the myocarditis incidence is a bit less after the third and after the second injection. That might have to do with immune tolerance. I'm not going to go into that today, but this is also something that must be expected with mRNA vaccines and doesn't really help their protective function. Okay, so here we have some myocarditis under the microscope. Okay, so on the left-hand side for comparison, we have some nice, healthy, straight heart muscle fibers that run from the bottom left to the top right. And on the right-hand side, we see vaccine-induced myocarditis. So this is a COVID vaccine patient who died and whose autopsy materials were reviewed by pathologist Professor Arne Burkhardt in Germany. And so he found severe myocarditis, so the heart muscle fibers are undergoing disintegration. Some scar tissue is seen at the top right. These more pale staining cells, these are fibrous cells, connective tissue cells. And these numerous small round blue ones, these are the lymphocytes which are in the process of attacking those heart muscle cells. So this is just the mechanism which I pointed out by way of cartoon earlier on. Myocarditis leaves scars, so the bright spot here in the middle, this is a fibrous scar. The red is remaining muscle tissue, and this is also from Professor Burkhardt's collection. And the scars within a muscle, heart muscle, they actually can act like rocks in the surf, you know, so they are acted as wave breakers. So the heart excitation occurs in the form of an electric wave that spreads in a very regular way from one trigger center, the sinus node, across the entire remainder of the heart muscle. And if you have a scar just within the path of this wave, then you can imagine that this electric wave can sort of get derailed, so the timing of the excitation and the path it takes through the heart muscle can be thrown off, and this can lead to arrhythmias, and this is more likely and more dangerous and higher heart rates, and this is a plausible explanation for the increased incidence of sudden cardiac arrest on the sports field, right, that we have witnessed the last couple of years. So these people, they may have had myocarditis that may or may not have, where was I?
The functioning of the heart and the implications of that scar that was detected.
Yes. Right,
creates the
right. So
arrhythmia
yes,
creates the heart attacks.
Yeah, yes, thank you. Yes, so these young people who suddenly collapsed while doing sports, they might have had myocarditis that may have healed, but with scars, and then these scars may have caused this delayed effect. Okay, so here we have lung tissue. On the left-hand side, some healthy lung tissue. The white areas are the air-filled spaces, and on the right-hand side, you see a big cluster of lymphocytes infiltrating that lung. You also see reduced amount of air-filled space, and also some of that space taken up by the lymphocytes. So this pneumonitis also observed by Professor Burkhardt in his case series. And here we have a thyroid gland on the left-hand side, again, normal, nice thyroid gland tissue with these beautiful follicles, and on the right-hand side, the entire structure is shot and everything is taken with scar tissue and lymphocytes.
I'm just wondering, are all
Yeah.
of these slides showing us post-mRNA injection tissues?
Yes, these are all post-COVID. Some of them are, actually the first one is after a adenovirus vector vaccine. This first myocarditis slide, I picked that one because it's particularly easy to see, I think, even for a non-expert. But very similar findings were also observed repeatedly after mRNA vaccines. So Professor Burkhardt, I'm not sure whether you have heard about him, he reviewed several dozen of autopsy cases, right? So these patients had died after vaccination. The autopsies were actually carried out typically at the hospital in question, and then there was no finding of vaccine damage. The families in those cases were not satisfied with those findings, and they approached Professor Burkhardt for a second opinion. Professor Burkhardt requested the materials from those hospitals, got many of them, not all of them, but many of them, and reviewed them again, and he went actually over these things in much more detail. For example, if you can really see many things only under the microscope.
Right.
If you just cut up the organs and you just look at them, then you can have some findings, but it is just simply not informative enough. And so he went at it very diligently, made histological, right, microscopic slides from everything, and then he found in the great majority of his cases, he found likely or very likely causal relationship with those vaccinations. And the recurring motive, this is why I'm bringing up these slides here, a recurring motive, he actually called this lymphocyte amok. So he says he had never seen before so many organs at the same time attacked by lymphocytes. This was a recurring motive. And one interesting finding too was that he found, for example, myocarditis, not only among young people. I mean, most of the cases he reviewed were actually old people. In many of them, he also found evidence of myocarditis. And I'm wondering how many of the cases that are reported as heart attack, for example, in the database, in the VS database, are indeed myocarditis, because simply from a sudden heart death, I mean, in an elderly person like myself, they would simply leap to the conclusion it was a heart attack, but it might have been a myocarditis in quite a few of these cases too. He also found mixed cases, so he found cases in which there was a real heart attack with vascular damage in the clot and myocarditis at the same time. Okay. And what he also found, I mean, he found typically organ damage not only in the particular organ that was the most implicated as the cause of death, but also at the same time in many others. So he found, for example, if there was heart attack, he still found something like this in the thyroid, or he found inflammation in the brain, or he found it in the lungs, and so on and so forth, in the kidneys. So the picture was really quite similar in many cases to a systemic autoimmune disease. So, for example, lupus erythematosus is a very well-known, distinct autoimmune disease that can also attack many organs at the same time. And this had really some similarity to that. Organ distribution may have been a bit different, but that probably has to do with the organ distribution of the vaccine, right? The vaccine has some preferences for going to certain organs. Okay, so here we have an inflammation of small blood vessels in the brain. In both cases, we have, again, these are cross-sections of those vessels. We see, again, these round blue lymphocytes which have invaded the vessel walls. And on the right, we actually see a small blood clot obstructing the lumen of this blood vessel. Here we have some larger blood vessels. These are from a different pathologist, Ute Krüger. On the left-hand side, a normal blood vessel with regular, smooth muscle structure in the vessel wall, and then in the middle panel, we have a clearly moth-eaten appearance of of the vessel wall, so it is disintegrating due to the vasculitis, due to the inflammation. On the right-hand side, also vasculitis and again a blood clot filling the lumen of the blood vessel. Here we have a study by another pathologist, Michael Merz from Dresden. He published this study in 2022. This patient actually had Parkinson's disease and some other diseases in his history, and he got 3 vaccinations, developed encephalitis with epileptic seizures and so on, and he was autopsied afterwards. Dr. Merz developed a simple yet ingenious assay to prove that the spike protein, which was expressed in the cells in the brain of this patient, was indeed due to vaccination and not to the virus infection, simply by testing for another viral protein, the nucleocapsid. Now, in a virus infection, all viral proteins should be present, including also the nucleocapsid. On the other hand, the vaccine encodes only the spike protein. So if you see only the spike protein but no other viral protein, that clearly points to the vaccine as the cause. In keeping with this conclusion, this patient did not have a history of a COVID infection, but he had received 3 vaccinations. 2 of these, the second and the third, were mRNA vaccinations. On the right-hand side, number 1 highlights a dead nerve cell, and number 2 highlights one of several lymphocytes in the brain tissue. Okay, so this points out, I think, that these examples should illustrate that indeed this mechanism of immune attack on cells that produce the antigen fits quite well with the pathological evidence. I find that important. I mean, pathology is always a key method for deciding on disease mechanisms. Now, there are some other plausible mechanisms for vaccine damage, as well as some implausible ones. For example, number 5, graphene oxide. I know physicists from Vienna who have looked for graphene oxide in various vaccine samples but have not found it. So I don't think there's anything to it. I have not seen any clear-cut evidence of that. The other 4 mechanisms listed here, number 1 through 4, they actually do have evidence to support them. The first is the one that we have already discussed, and we have direct spike protein toxicity, which cannot be waved away. There are several toxic activities, and they have probably been discussed by other witnesses here, so I'm not going to belabor them too much. I mean, they can go to the nucleus and cause mutations. Spike protein can bind to the ACE2 receptor and mess with inflammation and blood clotting and so on. So that is real. Then we also have chemical toxicity of lipid nanoparticles to consider, right? These lipid nanoparticles are designed to traverse cell membranes, so in a sense to break them up. And so this damage to cell membranes is real and can also cause downstream effects such as inflammation and so on. And then also, beginning in 2023, it was reported by many groups independently that the vaccines contained a residue of DNA from the production process, which increases the risk of genetic mutations. Now, can we figure out which of these mechanisms is the most decisive one? One clue really comes from the adverse event frequency after repeat injection. What should we expect in each case and what is observed? I already pointed out the reasons why with an immune response to spike protein or to any other antigen, in fact, we should expect an increased severity of adverse events after repeat injection. And now with direct spike protein toxicity, the antibodies that are formed by the first injection should actually bind and inactivate the spike protein. So what we should expect, if the spike protein direct toxicity were dominant, we should actually see fewer events, fewer adverse events after the second injection than after the first. With chemical toxicity and DNA toxicity, I see no reason to expect any change at all, so I would expect similar adverse event frequencies after first and second and repeated injections. So the only mechanism that fits the observed pattern of increased adverse event frequency after the second injection is the immune response to spike protein, which also fits best with the pathological evidence. Okay, so I have already summarized this. I don't have to belabor it, I guess. So it the most important mechanism seems to be indeed the immune response. Now, why is it important? It is important because the this mechanism is completely general. It depends only on the foreign nature of the antigen encoded by an mRNA vaccine. It doesn't matter whether it's a COVID spike protein, or it's a measles virus surface protein, or it's an RSV virus surface protein. They will all be foreign antigens. They will all set off the immune system, and so they will all cause the immune system to inflict cell and organ damage by destroying the cells that have taken up the mRNA vaccine particles. And as I mentioned earlier, this uptake cannot be prevented by pre-existing immunity. These vaccines will get there, they will get the man, so to speak, they will cause the damage. This must be understood as an intrinsic, fundamental flaw of the mRNA vaccine technology. That's my point. That's really the key point I want to make with this entire presentation. The damage that we see, that we have seen after the COVID mRNA vaccines, must be expected to recur in a similar way with similar severity after any and all future mRNA vaccines against other viruses. That's my key point.
Okay, we've just on that, for the vast unwashed, as am I and most of us, we've heard the term self-replicating.
Yes.
And now your last comment refers to the consequences of further injections, but as I understand the concept of self-replicating and the concerns expressed by some other experts,
yes,
what are your thoughts on that?
Yeah. Well, it seems that I asked a Japanese doctor. Right? Japan was the first country to approve one of the self-replicating vaccines. Apparently, uptake has been very low. So in the was it the third or the fourth quarter in one of the final quarters of 2024, only 15,000 injections had been distributed in Japan. But it was a significant number of adverse events reports, and the frequency of these adverse events increased relative to the previous, if you want to call them, conventional non-replicating mRNA vaccines. So empirical data indicate already that they are risky. Now, there are many reasons why that would be so, right? If you have a conventional inactivated virus vaccines, one upside of that is that you know exactly how much antigen you are going to inject. You know the dose exactly. If you inject an mRNA vaccines, there are many variables. How effectively is it distributed throughout the body? How is it? How many cells take it up? How much how efficiently will they express the antigen? So you have to actually expect a fairly large variation in the level of antigen expression. Now, with the self-replicating mRNA vaccines, you're adding another unknown. You're injecting a small starting amount of the mRNA, and then it starts replicating. We don't know how many times it replicates. If it undergoes 10 replication cycles in one person and it undergoes 20 in the other, then the second person has 1,000 times more than the first. Okay, so you can have a huge variation in the amount of mRNA that's formed, and then this can again be turned with varying efficiency into protein. So you must expect a huge spread. You must expect a huge spread of antigen expression. Some patients might really end up replicating the mRNA very effectively and then also expressing the antigen very effectively. So very many cells will then come under attack by the immune system and will be destroyed. And the fundamental problem really remains the same as well, right? So this mRNA, to the extent it spreads within the body, can really not be intercepted effectively by the immune system because it spreads without a capsid. It spreads within probably exosomes, which are membrane vesicles just simply budding off from the body cells. They will not be recognized as foreign. They will not be intercepted by the immune system.
Okay, that's helpful. I I do note the time. You've covered so much that it's worthy of further questioning, but I'm going to now turn you over to the commissioners who I suspect have a few questions of you. Oh, I should also say, just before you start, my friends, your PowerPoint will be an exhibit. It will be available on the NCI website along with your testimony. So I know it's got a number of references, background references for the data that you've produced. That'll all be available online through this exhibit.
Okay.
So my friends will now ask you some questions.
Hi, Dr. Palmer. I'm not a scientific, so for me it's hard to understand all of this. But tell me if my basic understanding is okay, and then I'll have one question for you. So as I understand it, the COVID vaccine has three elements: the lipid nanoparticle, the spike protein, and the mRNA. The lipid nanoparticle serves for biodistribution into the cells. And then the spike protein is to have an antigen response. And the mRNA role is to reproduce the spike protein. Is that okay?
The mRNA provides simply the blueprint, it's the instructions for producing for the cell, right? So the cell gets hold of the mRNA and starts reading it and then translating that information into an actual protein, right? This is the same way it also works with the cell's own protein synthesis. So in that case, the mRNA is produced in the cell nucleus, is then transferred, right, as a copy of the DNA sequence. Then the mRNA is taken to the cytoplasm outside the nucleus. Then the so-called ribosomes attach to this mRNA, reading it, and are just assembling as they go. They assemble the protein. So they translate the information of the mRNA into the information of the protein sequence.
Okay, so the first question of your PowerPoint was, is the toxicity inherent in the mRNA platform itself?
Right.
So I saw in your PowerPoint that you showed that the cells were not recognizing the lipid nanoparticles.
The antibodies are not recognizing.
Yeah, so they were not able to react. And I saw also from the picture you had that the spike protein was like expanding and creating problems. But I'm not sure I understand the clear response to your question, the toxicity inherent in the mRNA itself.
Okay, it's not the mRNA molecule as such, but it is the mRNA platform. It is the general idea of introducing mRNA packaged in synthetic lipids as vaccines. That is the problem. So the mRNA vaccine paradigm, that's the problem. It's not literally the mRNA molecule as such.
Okay, thanks.
Thanks very much. Very interesting presentation. I think it's fair to say that by now we have a vastly overwhelming amount of information on the toxicity of these vaccines. Most of it from the peer-reviewed literature, some of it from the epidemiology. Why is it that there is still a significant reluctance amongst many people who work in the field to accept that these things are dangerous?
Well, it's not a scientific question. I'm not an expert in that regard. It's anybody's guess, really. Why people want to see no evil? I don't know why they don't.
Thank you for your presentation. I'm really interested in this graphene oxide.
Okay.
Why is that present? Why would they have it there?
Well, as far as I understand, they don't. I have not seen, excuse me, I have not seen convincing evidence of graphene oxide in vaccine samples.
So that's why you had the question mark.
That's the question mark. Yeah, I wanted to, just as an example of, I mean, there are several valid damage mechanisms, but there are also several invalid ones. And I think these stories about the invalid ones, I think they are put out on purpose to distract and confuse people.
Okay. My one other question. I have a niece she's had 4 miscarriages. And they have been testing her killer cells,
Okay.
which are highly, highly elevated, apparently, in most of the girls that are spontaneous aborting. And they're now trying lipid infusions in the UK. How would that work? Would it neutralize the T killer cells?
I would have to know more about the particular lipids. I mean, lipid is a very general term, and I cannot.
Yeah. So it's still experimental.
Honestly, I don't know the method at all.
Yeah.
I cannot really.
Okay. Thank you.
Anybody else? Vincent? Let me ask you a couple more, if we can. I know you're a scientist and not a politician, so you're not going to be exploring in any depth what the motivation might be behind others in your field or public health agencies. But the fact that you've been able to review this with us in the way you have, indicating what certainly appears to be a very compelling concern about the safety of this product.
Oh yeah, I mean,
and the fact that we're addressing it here at the NCI, but it has been ignored by almost the entire mainstream press and
that's right.
Political institutions. So my question is this: you've shown us some slides of investigations done by pathologists,
Yeah.
microscopically examining tissues from post-COVID mRNA injected fatalities, indicating the damage to the tissues that you've shown us.
Right.
There's a pathologist named Kruger, another one named Mertz, and perhaps others.
Burkhardt.
And yes.
Mm-hmm.
So unless you look for something, you don't find it.
Very, very true.
You know where I'm going. So the pathologists who've actually microscopically examined the tissues to identify the damage that you've now described, now, my understanding is there wasn't any sort of systemic approach to the investigation of tissues in the vast majority of people, notwithstanding these huge spikes in excess deaths and other conditions following the vaccines. Am I right that the slides that you've shown us have been generated by a very small portion of people who were able to investigate these things? And
it is true.
Tell us about that if you can.
Well, okay. So I mentioned Professor Burkhardt that he was approached by the families of people having died after vaccination to review those autopsies. Now, Professor Burkhardt was actually he was in his late 70s. He's actually deceased now. So he actually wanted to delegate that work to some colleagues of his that were still active. And those colleagues initially agreed, and then they suddenly didn't agree and just said, leave me alone. So it seems very much as if these people had been under pressure to not carry out these investigations, which was the reason why he then took it upon himself to spend his final years actually doing these kinds of reviews. Michael Mertz is a pathologist. He's employed at the City Hospital of Dresden, so he's actually an active one. And apparently his boss, who herself doesn't really make public appearances, was at least decent enough to let him do his work and not interfering with it, without interfering with it. But indeed, there is a shortage of such reports. What I do find remarkable is that very many of the pathology studies, of the better ones, have come from Japan. Japanese physicians still seem to have, at least at a higher percentage of them, still seem to have traditional ethical principles and follow them. That I find remarkable.
Just to finish off then, we've seen what appears to be almost a systemic disinterest or disregard to the kinds of risks you've identified. And even to this date, the mainstream medical community is not going into the investigations that you've shown us some of. It's still not happening, even though the information you've brought to our attention is well known within your scientific community, right?
The first time I discussed these fundamental problems was when Ted Kuntz had me on in a presentation in late 2020. That was before the first injection was administered. You really only have to put 2 and 2 together in order to find the risk. And I discussed the same question then with my pharma boss, Sucharit Bhakti. We hadn't been in touch for a long time, but after that we got back in touch, and he thought exactly the same. He made the same prediction already before the first shots were administered. So it's textbook knowledge. You simply have to analyze your own textbook knowledge.
And even though historically vaccines pre-mRNA technology, but the traditional vaccines, when they indicated risks that were not nearly as significant as those shown on VAERS and the data you've produced, not nearly as significant as those, they were shut down because the public health authorities recognized those risks. They were shut down. But there's been no shutdown of the mRNA.
That's right. But I mean, in those cases, one must say risks are also pretty frequently denied and downplayed and this sort of thing. Discovery of those risks is avoided by not carrying out studies with proper controls and so on. So.
But at least the public health authorities pulled those products.
Yeah, indeed. I mean, it wasn't all great before, but of course the problems that we are facing now are on a different order of magnitude,
Correct. Well, subject to any questions from the commissioners, those are my questions. And on behalf of the NCI, Dr. Palmer, we are immensely grateful for your contribution and your work in this area and your courage for speaking out as you have today and previously. So
thank you very much for the opportunity. I appreciate it.
Thank you very much.